Okay, in this particular lecture, we get to actually apply the cell cycle and what issues might arise when we talk about cell cycle in cancer. So our essential question will be, what factors affect the cell cycle? And we can also add in, let's see, we'll do a separate one about what causes cancer. Okay. So the cells within an organism's body divide and develop at different rates. It just depends. So skin cells are dividing rapidly all the time. Brain cells don't particularly divide rapidly. Sometimes not at all. You know, they can, but they're real slow. So different cells divide at different rates. Cell division, so that whole process that we talked about that includes interphase, and then when you get to mitosis, it's PMAT, that whole thing is dependent and controlled by these three things. One we call anchorage dependence, one we call density dependent inhibition, and, oh, sorry, I meant four things. Whether nutrients are there, and then also growth factors. So anchorage dependence is the need for cells to be in contact with a solid surface. So they have to have it's kind of like a house. They have to have a solid foundation before you can build the house. So before we can really put cells on it, we need to make sure that we have like a solid surface for them to divide on. Density-dependent inhibition is when crowded cells stop dividing. So basically cells, if they're touching on all sides, like this guy, it's touching here, they're on the base, they're touching on both sides, it says no division. It's not dead, it's just meaning no division. Okay. If there's something happens where the cell does die, well then that creates a space where we can then, one of these cells will divide. They don't go upwards, they only go side to side on that kind of basement thing. I'll show you that here in just a second. But when they're crowded, they stop dividing. If you don't have the right nutrients, they won't divide. Growth factors, certain things stimulate cell division. If those aren't present, it won't divide, or if they are present, sometimes we divide too much. So here's anchorage. This is kind of a little petri dish of sorts. As long as they have a spot on the bottom, then they're available to divide. Kind of like this guy back here. Single layer of cells, if we have that whole layer, that's density-dependent inhibition. Everybody's touching on all sides, so there's signals for us to do no division. However, if we get a little scupula in here and we take a couple of cells out, well, then that will trigger cells around it to divide to fill the space. And there's lots of communication. That's why we talk about plasmodesmata and other different kind of what is the word? Like channels between these things to be able to make sure that they can communicate, so they put the right number of cells back in the middle. Okay. Cancer cells were formed by overlapping, and they don't follow those particular rules. All right. So some other things to look at: addition of growth factor. If the growth factor is not there, the cells will not divide. If we add that growth factor, then the cells will begin to divide. So we need a growth factor. That's all that this little thing says. Okay, so no dividing over here, but dividing with a growth factor. So the cell cycle control system is a cycling set of molecules in the cell that triggers and coordinates key events in the cell cycle. Okay? It tells us that we need to follow these rules. There's very specific rules. There aren't many, but there are enough that have to be followed. Okay? So we've got some chemicals that help make sure this happens. So there are checkpoints in the cell cycle that can stop an event or signal an event to proceed. So we got a couple of checkpoints. So here are the three major checkpoints in the cell cycle. We have one at G1. which will allow entry into the S phase. And remember, we talked about the S phase. So the S phase is, right, DNA duplication. So at this point, if you're duplicating the DNA, then you're trying to say, all right, cell division is going to happen. You're doing that. Okay. But if we're not ready to do the cell division, if we're not moving in that direction, it'll actually cause the cell to leave the cycle, and it enters this kind of non-dividing G0 or G0 phase, where we just say, no, 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 we're not dividing, we don't need to do anything. It's like a pause phase. This is probably where we would find most of your brain cells. They don't need to kind of divide. They're there, they are established as an adult, and so you don't need to do anything with those. So they're not going to start dividing. Okay, there is a G2 checkpoint, so remember G2 leads into mitosis. Actually, I don't think that's quite the right one, but basically we're saying, okay, we're ready, we got S phase together, we're going to move on. The M checkpoint just says we are ready, in fact, to undergo mitosis, everything's clear, everything looks good. So because mitosis is a fairly complicated process with a lot of moving pieces, we want to check it several times and make sure that we're right. If it's not right, the cell can undergo this thing called apoptosis, where it just kills the cell off and says you're not dividing properly, let's go ahead and start over. So we really need all these checkpoints in place to make sure that everything's proceeding as it should. So research on the controls of the cell cycle is one of the hottest areas in biology today because of how it relates to cancer. Okay, so if we can sort of figure this out, then we can work through kind of some cancer solutions. So here's kind of the control system we talk about. We're in interphase. Remember, all of the brown is interphase. If you don't remember, brown is interphase here. Right? The blue stuff is mitosis. Okay. So we enter the G1 phase. If we're deciding at this first checkpoint right here that we don't need to divide, we'll enter into the G0 phase that says, all right, we're just hanging out. If we pass this checkpoint and we get the green light instead, we'll move into the S phase. And it'll begin the whole process. There we go. That's where they're at. It'll begin the whole process. We get through G2 and it will check, are we ready in fact to build 2 new cells? If we get through the G2 checkpoint and we are ready to build 2 new cells, then great, we can move into mitosis. Somewhere along the way there are mitosis checkpoints also, just to ensure that the whole thing is moving along smoothly and we're getting 2 proper new cells. So these are our 3 major checkpoints that we would look at. Okay. And there's growth factors involved in this whole thing. That's the whole point, is again, growth factors would be involved here to tell this thing to move to the next checkpoint. So we've got growth factors, as we mentioned in the beginning, will signal it, will chemically signal for this thing to move past that checkpoint and out of the G0 or G0 phase. So cancer currently claims the lives of 20% of the people in the U.S. Okay. Cancer cells escape controls of the cell cycle. So there are very specific rules: the anchorage dependence, the density dependent inhibition, the growth factors, nutrients. So all of those things are really rules, and cancer doesn't follow those rules. So cancer cells will divide excessively and invade other tissues of the body. So it's just, cancer really is just uncontrolled cell division. That's kind of our basic definition here. Okay. Uncontrolled cell division. A tumor is a mass of abnormally growing cells within otherwise normal tissue. So it's kind of uncontrolled, abnormally growing cells. We have 2 types. We have benign tumors and we have malignant tumors. So benign tumors remain at the original size but may disrupt certain organs if they grow in size. So they kind of stay in the original tissue site. So usually these things can be removed fairly easily because they're within a space of tissue. So they're staying at their original site, they're in a certain location. For the most part, we can remove those. Malignant tumors can spread to other locations in a process called metastasis. So that when a cancer becomes metastatic, it is spreading to other parts of the body. That's usually stage 4 of cancer. Okay. An individual with malignant tumor is what we say has cancer. Okay. There are levels and stages of how these work, but it all originates from abnormal and uncontrolled cell growth. So here might be what it looks like if we've got some cancerous cells in a healthy tissue right about here. What they'll end up doing is they'll continue to grow and divide uncontrolled and unchecked, even though you can see over here there's no room for them. They may or may not be getting proper nutrients. They're, they don't have any space. They're not doing the kind of density dependent kind of inhibition. They don't have an actual basement membrane to grow from, so they're just growing wildly here. At a certain point, when they really become metastatic, the cancer builds large enough that it'll actually touch these blood vessels. And so they actually get around the whole thing of not having nutrients by getting the blood vessels and spreading out to certain places and rerouting blood vessels so that they can continue to feed themselves. Cancer is a very rogue thing. It just doesn't follow rules, and it does whatever it wants to, and it's ugly. Okay, so here's a good one. Cancers are named according to the organ or tissue in which they originate. So 2 cases in my family, my grandmother actually had cancer of unknown origin, which is very rare, as they never knew exactly where it came from. My dad had what's called squamous cell carcinoma, which means that it originated in the squamous cells of the lungs, and it was a carcinoma because it was within an internal body covering. So that's a carcinoma. It was both the type of cell and the type of covering, or the type of tissue. So they called it a squamous cell carcinoma. Leukemia originates from immature white blood cells within the blood or bone marrow. So as a connection and as a to kind of investigate a little bit further, I'd like you to find maybe 2 other types of cancers, give kind of more of their scientific name other than carcinoma and leukemia. See what you can find and give me 2 examples of cancers and give me where they would originate. So you're going to have to do a tiny bit of research if you want 5 points towards the next test, but I don't think it'll take you too long. This lecture won't be too long. So that's your little extra, kind of your Easter egg today, is to find 2 other types of cancers and where they originate. Give me the names. All right. Localized tumors can be removed surgically or treated with concentrated beams of high energy radiation. You just shoot the crap out of them with radiation and they, disappear. So you can do that with localized tumors. Unfortunately, with metastatic tumors, they're treated with chemotherapy, which is an insane process that really eliminates all cells, both cancerous and healthy. And really, the growth rates between the 2 are a little bit different, so they hope to kind of build the healthy cells while removing a whole bunch of cells. So it can make people very, very sick depending on the chemotherapy. Though I will say, and they do kind of a treatment, personalizing cancer treatment. There are also lots of things like immunotherapies are a big thing now, where they're actually finding that they can use parts of the immune system in order to help kind of boost cancer, because the body will fight cancer already. The body doesn't enjoy cancer. It's just sometimes cancer can go unchecked and out of control, and it develops at a faster rate than the body can handle. But there are some immunotherapies that boost the immune system to be able to find these guys and take them out. But we can have some personalized ones that look for sequencing for genome tumor cells and tailoring treatment based on a person's specific genetic profile. What types of things might be there that would lead to cancer? Okay. Another example, well, maybe we might have some here, but this might be a good I've gone a little long, so this might be a good place to take a break and then come right back if you need to. So more than 100,000 Americans are stricken by cancer of the colon this year. Colon cancer is one of the best understood types of human cancer and illustrates the principle of how cancer develops. So I really wanted to show you this. We're not going to go into all the details, but there are things called oncogenes, which are cancer-causing genes. We'll have some different mutations that cause growth of a small tumor, and then there will be additional mutations as these things grow and develop, which we'll touch on later whenever we go through the genetics side of things and talk about DNA a little bit more that way. But you see there's some cell division that's increasing once an oncogene, or which is really just a cancer-causing gene, is activated. It's really all that it means. And so that starts to begin to develop and grow and divide out of control. Okay. Then tumor suppressor gene is inactivated. So you have these genes that suppress a tumor that say, no, 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 you don't do that. And they go and get it. But the cancer will also have an inactivation of things that would suppress a tumor. So we have a benign tumor that's there in that particular location. And then as you move on, a second tumor suppressor gene is inactivated. So we totally had a system in place for both of these things that got inactivated. And so it became a growth of a malignant tumor where it starts to take over blood vessels and it reaches out to other tissues and parts of the body. It's a nasty thing, man. Just follow the rules. That's all the body wants it to do. Follow the rules. It'll be all right. But after heart disease, cancer really is the second leading cause of deaths in most industrialized nations. As what happens with longer lifespans, there's more time for mutations, and those are really what would cause cancer. Cancer can run in families of individuals who might inherit an oncogene, a cancer-causing gene, or a mutant allele of a tumor suppressor gene, make it one step closer. But most cancers can't be associated with an inherited mutation. Most of them are not inherited per se, it's just as those things come up. You might have a greater prevalence. I've had cancer on both sides of my family for various reasons, so it is worth looking into to see if my family, me and my future family, contain oncogenes or some mutant tumor suppressor genes. It's worth looking into, but most cancers are they come up not from mom or dad. It's just a part of what happens in the body. Okay. Carcinogens are cancer-causing agents that alter DNA. Most mutagens are substances that promote mutations or carcinogens. Look at the words out. I worked with one called, God, what was that thing called? Mishmayeski and I both worked with it. Oh, it wasn't methylene blue. Oh, it's something else. I'll have to ask her. We'll have to figure it out together. But it's a nasty thing that we used for DNA, being able to find DNA stuff. So whenever you look at DNA, we both used it, and I worked in a highly kind of used area of that. She, I think, maybe got it. I don't know, she touched it or something. So we're both just like, well, I hope we don't get cancer from being in those kinds of areas. But it was definitely a mutagen, and it would promote mutations. So we'll all cross our fingers and think happy thoughts that maybe it won't do it. That is the nature of science. We make sacrifices, and it's our own fault. Two of the most potent carcinogens and mutagens are X-rays and UV radiation from sunlight. So X-rays they didn't know for a long time, and UV from sunlight, so just make sure that you're taking proper care to wear sunscreen when you're outside. Don't get big sunburns. I don't mess around with that. I'm old and I put sunscreen on all. If I'm out, it's always there. So one substance known to cause more cases and types of cancer than any other single agent is tobacco. More people die of lung cancer than any other form of lung cancer, and this is a true statement. So please don't mess with that stuff. This is my public service announcement. This is what got my father was 40 years of heavy smoking, a pack a day, 40 years. Just they're just not cool and they're ugly, and it means that it puts you at a higher risk for lung cancer. And I know that was my dad's biggest regret. So stay away from tobacco. Don't ever start, don't ever mess with that stuff. Okay? So most tobacco-related cancers come from smoking. Passive inhalation from secondhand smoke also puts people at risk. So smokers can affect themselves and others, which is a bummer. Tobacco use, sometimes in combination with alcohol consumption, causes cancers in addition to lung cancer. It's just, it's not a good thing. So I have to put it out there. Don't smoke. Okay? Don't do it. Oh, look, here are some other things that you might look up that might be helpful for that thing that I asked you to do. But here are some other cancers and how many new cases we have. Notice we have a ton of prostate cancers, breast cancer, lung cancer, and colon cancer. It goes down from there, but there's still a ton of cancers. And you notice their risk factors. A lot of it's tobacco smoke, tobacco smoke, tobacco smoke, tobacco smoke. So just, really have to just avoid that one to reduce our risks of that and try to, let other people know. So that's where we end this particular section. You won't have another lecture before you get to, test time. But make sure that you review. If you have any questions on this or any of the previous material, send me an email or come by tutoring or flex time and let's figure it out.